Alright, hey yo, how's it going? Today we are going to be talking about DNA replication, which takes place during the synthesis phase of the cell cycle. So if you haven't checked it out already, you should go check out my video on the cell cycle. Hopefully you won't hear me chewing in that one or this one, because rumor has it you could hear me eating some food in one of my videos. I'm sorry about that. Embarrassed, mistakes happen. So first, before we talk about DNA replication and how it occurs, we first want to talk about what it is. So DNA replication is just what it sounds like. It's going to be copying the DNA to make an identical copy. And the whole purpose of that is so that both of the offspring cells have the same DNA when they go through cell division. We want both cells to have the same set of instructions. DNA holds those instructions. So what we're going to end up doing is we're going to end up taking one DNA molecule and creating two identical copies of that DNA molecule. So how we're going to approach this is I'm going to take you step by step through what each step is when we're doing DNA replication. So what we have to do when we replicate DNA is we actually have to get in here into our molecule of DNA, and we actually have to copy the nitrogen bases. Remember, the nitrogen bases are kind of the step part of our ladder. We need to copy those in order to bring in new nucleotides and build new strands. We start off with two things called the original or template strands of DNA. All right. What template means something we're going to copy off of. So these guys are going to be what we're going to copy off of, what we're going to build off of. So the problem is, if we're trying to come in here and copy the DNA, we keep getting blocked by that sugar phosphate backbone, and we can't get in there and copy that DNA. So the first step of DNA replication is going to be to unwind the DNA double helix. So we're going to make it into regular ladder form. So step one is to unwind the DNA. There's a molecule that does that for us. So an actual molecule will come in here and will swoop in, much like my little lightsaber here. Oop, no, just one of them. And what it's going to do is it's going to take the DNA and it's going to go nope. And it's going to unwind the DNA so that it's into ladder form. That molecule that unwinds the DNA is something called DNA helicase. And how I always remember this, and I've been told since that this is kind of a goofy way to remember it, is that helicase sounds like helicopter, and helicopters go woo and spin like their top propeller, just like you would have to spin the DNA in order to unwind it. Once our DNA is unwound, we've got to move on to step 2. So now that we have some unwound DNA, Now that we have some unwound DNA, our next step is going to be to get in and actually be able to copy those nucleotides. In order for us to get in there and copy the nucleotides, we're going to have to break apart the nitrogen bases. So we need something to come in here and to kind of karate chop the nitrogen bases. So guess who gets to come in again? That's right, DNA helicase. The DNA helicase comes in and it goes cha 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 cha, and it slowly breaks those hydrogen bonds in between the nitrogen bases, which allow us to then unzip the DNA. So now that our DNA is unzipped here, as you can see, it's only actually partially unzipped. So DNA helicase doesn't want to go clear up to the top and completely unzip our DNA. The problem with completely unzipping our DNA is it's actually 4 billion base pairs long. So 4 billions of these letters paired up. which means it's super long. If it unzipped the whole thing at once, it would just make a big mess, and the DNA would fall apart into 2 strands, and it'd be like super complicated to copy. So DNA helicase only unzips a little bit at a time. How it unzips again is it breaks through those hydrogen bonds. Okay, now that we've got the DNA unzipped, we're ready to start building the new DNA strands. So these new DNA strands are going to be built off those original or template strands, which remember are in green on your screen right now. Fortunately for you, floating around in your cells like alphabet soup or something, there are free-floating nucleotides. Those free-floating nucleotides include adenine, cytosine, guanine, and thymine. Our cell has a molecule that will come in and it will grab one of the free-floating nucleotides, and hold on, it's going to take me a minute to get this right. Okay, let's try again here. So it has a molecule, your cell has a molecule that will come in and it will grab the free-floating nucleotides and it will move it down to our original strands. Move it right next to it so that these molecules, these nucleotides, can start to build the new strand. And as you can see in my pictures, as usual, not awesome at art. It was never my greatest strength, although I'm really good at drawing trees. But this molecule moves these nucleotides down and puts them in right next to the original strand. And that one's not going to shrink for me very well. Nope, I thought I could get that smaller. That one's a little ugly, but that's alright. It's a C and a G right next to each other. And then what that molecule does is it pastes those molecules in by building the backbone and connecting the backbone between them. And it pastes those molecules in. So now we have a T across from an A again, a C across from a G again, a G across from a C again, a G across from a C, and then on the opposite side we have the exact same thing, just flipped. At this point, the cell has just rebuilt the DNA right up, clear as far up as we can to the point where it's been unzipped to. So we'll need DNA helicase to come in and unzip more at this point. At the farthest point up, the farthest point we've unzipped to, it'd actually be up here, not down there. The furthest point that we've unzipped to, that is called the replication fork. Right? Not fork like what you eat, but like fork in the road. So fork out of the DNA there. Right? And that, like I said, is the furthest place that we've actually unzipped our DNA to and been able to copy up to. So what would happen would be DNA helicase then would move on in and would undo the next few hydrogen bonds, and then we would rebuild more DNA right next to those original strands. The molecule that rebuilds the original strands is something called DNA polymerase. If you notice, both DNA polymerase and DNA helicase end in ASE. Any molecule that ends in ASE is going to be an enzyme. Just like catalase, the lab we did with catalase that ended in ASE, that is an enzyme. So DNA polymerase is going to be an enzyme that speeds up the building of DNA next to the original strands. Helicase unwinds and unzips. Polymerase builds. This whole process keeps going on. DNA helicase comes in, unzips a little bit. DNA polymerase builds the new DNA right off of the 2 template strands or the 2 original strands. DNA helicase and unzips a little bit more. DNA polymerase builds a little bit more. This keeps going till we finally reach the end of our DNA, and what we end up with is 2 identical daughter strands of DNA. If you look at these guys, they are identical to each other. So TTC, GGCA, and then over here you got TTC, GGCA, and then If you scroll back or rewind back, you'll see that that was identical to the DNA that we started with. So we got now 2 molecules of the exact same DNA. The whole goal there was so that we can pass the same exact DNA onto the new cell that we're getting ready to make. You can also see that we started with 2 green strands, right? So those were our original strands, the green ones. Well, now we have 2 blue strands that we're going to end up with. Those are our new strands. So we still have the 2 original strands, but since they went to different molecules, we see that DNA replicates in a semi-conservative way, meaning that the new molecules have 1 original strand and 1 new strand. If we were to copy these 2 again, we'd end up with 4 molecules of DNA. Only 2 of the strands would be green from the original one. 2 of them would be blue, and then the other 4 might be whatever color we chose, right? Obviously DNA is not colored, but we're using the color here to represent the original strand and the new strand. All this is going to happen during the synthesis phase of the DNA of the cell cycle. That happens right after G1, right before G2, which happens before mitosis. The whole purpose of synthesis is to copy that DNA to get ready for cell division. Alright, I've got one last goal today. We're going to talk about a couple different terms for DNA that's going to be essential for us figuring out mitosis, which is coming in the next video. So, all DNA, well, first of all, let's start off with all humans have a certain amount of DNA in their cell. Our DNA has been chopped up into 46 pieces of DNA. So you and I have 46 pieces of DNA in each of our cells. Those 46 pieces of DNA are different from each other. Each piece of DNA will always come in one of 2 forms, and it can change between the 2 forms. Each piece of DNA will always be either long and thin and unwound, like that piece right there. That piece is called chromatin. Chromatin is thin. Alright? Or our DNA will come coiled up. If it's coiled up, it's going to have a different name. An S is kind of coiled. So how you'll remember that one is it is chromosome. So chromatin is thin, chromosome is coiled. So let me give you a little analogy here. I give you a 100-yard piece of yarn, and I say you need to walk around with this piece of yarn all day, and I don't want you to get damaged because it's really important to me. That's going to be used for when I knit my grandma's sweater later on. So you can't mess up that DNA, or you can't mess up that yarn. Would you rather carry it around all day coiled up, or would you rather carry it around not coiled up? Hopefully you answered wound up. It's less likely that it's going to get damaged when you're walking around all day if it's wound up. Just like that, our DNA is less likely to get damaged when it condenses down and gets coiled up when it's moving. So our DNA will be in chromosome form when it's moving. When it's not moving, it will be in chromatin form. Well, in order for DNA to be able to move, there has to be no nucleus. So we'll see that in mitosis, at some points there's going to be a nucleus, some points there's not going to be a nucleus. If you can always look at it and say there's no nucleus here, then you're going to know that the DNA must be in chromosome form because it's got to be able to move. Similarly, if there is a nucleus, the DNA is not going to be moving, so it's going to be in chromatin form. Well, we just got done learning what happens during the synthesis phase before mitosis. During the synthesis phase, our DNA replicates or copies itself and makes an identical copy. So if we end up with an identical copy, it's going to look something like that. Now our DNA has our body has a little way that it keeps the DNA from getting mixed up, when it copies the DNA, it puts a little rubber band on it. That rubber band is called a centromere. Right? And that rubber band is to keep the two identical pieces of DNA together. Since those are two identical pieces of DNA, we can now call them sister chromatids. Alright, now this is where it gets confusing because we're talking about tids and tin and this and all that, and it all sounds the same. But those guys are sister chromatids because either side is identical to each other. However, DNA is always in chromatin or chromosome form, so the technical name for this one would be chromatin, because it's thin still, chromatid, because it's paired. And then this one, since it's coiled, would be, I'm trying to change it to white, chromosome. And because it's paired, chromatid. So tin is thin, some is coiled. It will always be one of those. If it's paired up, it's also a chromatid. Not too bad. Here's where it gets a little tougher though. This right here is one chromatin. This right here is one chromosome because I can pick that up. I could go like this and I can, not really. It's not going to let me do it, but if I could move the whole thing around because it's got a rubber band on it and it wouldn't come apart. However, since there's two copies of the same message of DNA there, this is actually, I'm not on the marker. This is actually two chromatids. 1, 2, because it's got two of the same message. And this is as well two chromatids, because it's got two of the same message. 1, 2. So it's one chromatin, two chromatids there. So the number of chromatids will always be double the number of chromosomes or chromatin. Let's practice. So here, let's say I've got that many pieces of DNA. Tell me how many somes, chromosomes, how many tins, chromatin, and how many tids you got there. Okay, first of all, somes means coiled. So is that coiled up? No, it's not. So then there's no somes there. It is thin like tin, so it is chromatin. There's 1, 2, 3, 4. So that's going to be 4 chromatin. They are not paired up, however, so that's going to be 0 chromatids. It's not 4 chromatids, it's 0, because they're not paired up. If they're not paired up, they're not chromatids. One last example. Right here we have how many somes, how many tins, and how many tids. So how many chromosomes, chromatin, and chromatids. There are 4 chromosomes there. 1, 2, 3, 4. Since it's coiled up. Since it's coiled up and not thin, there are 0 chromatins because tin is thin. And then lastly, there are 8 chromatids because each message or each number of instructions counts as a chromatid as long as they're paired up. So 1, 2, 3, 4, 5, 6, 7, 8. Alright? This is going to be essential for understanding our next video, which is mitosis. I'll see you then. Good luck. Good night.